Hello everyone, welcome back to Karan Academy. My name is Karan and today we're going to be going through anemias and hemoglobinopathies. Now anemias is a huge concept and there are a lot of subtypes and there's a lot to know. So hopefully today we'll be breaking this down into a systemic manner that's easy to digest, unlike much of the iron that we will talk about as well. And it's definitely something that's very high yield when it comes to exams. So yeah. This is a good overview that I've always found very useful, and it's a system that's very commonly used. So whenever you have an anemia, which by definition is a low hemoglobin, you can broadly classify that as either microcytic, normocytic, or macrocytic, depending on the mean corpuscular volume or the mean cell volume. Now, the MCV just refers to how big or small the cells are, and obviously that can be the micro, normo, or macrocytic. A good acronym that I found useful for each one of them is we have TAILS for microcytic, ABCDE for normocytic, and BOM HP. This one doesn't make sense for macrocytic. You don't need to know all of these, but the ones that are in red are definitely ones you need to know quite a bit about, and we'll talk to them as well. So let's start with the microcytic anemia. So in terms of its features, you'd have a low hemoglobin, relatively straightforward because you're anemic, right? And then you would have also a low MCV. Now the causes, as I mentioned, TAILS or thalassemia, anemia of chronic disease, iron deficiency, lead poisoning, and sideroblastic anemia. Before we go on to that, let's talk a bit about hemoglobin. Now hemoglobin is obviously a very important component of your blood, right? And you have about 270 million red blood cells, and you have about 270 million hemoglobin molecules per blood cell. And as you can imagine, that is a lot, right? So each hemoglobin structurally is made up of 4 globin subunits. And you can see here, we have 2 of each type. So we have alpha chain 1, alpha chain 2, beta chain 1, beta chain 2, right? So your 4 subunits, that consists of a polypeptide chain, which is a protein. And then you have a heme group, which actually contains the iron in it. The few important things you need to know, and I'll get to why these are important, is understanding the different types of hemoglobin. So we have HbA or HbA, which is composed of 2 alpha units and 2 beta units. And this makes up most of the hemoglobin in your body. It's about 98%. Then we have HbA2, which is 2 alpha and 2 delta subunits. And that makes up about 2 to 3%. And it's still found in adults. And then we have HbF or HbA fetal, which consists of mostly, so this is a bit different because it's not as commonly found and consists of 2 alpha and 2 gamma subunits. And it's obviously present in infants. HbF is special because it has a very high affinity for oxygen, which is kind of what you need in the neonatal periods before conception. However, since this obviously isn't needed in adulthood, you switch over to mostly HbA and some HbA2 soon after birth. Thalassemia is a disease affecting these subunits. So depending on which subunit is affected, we would call it a few different things. So with the thalassemia, we have alpha thalassemia and you have beta thalassemia. So with the alpha thalassemia, you are affecting the alpha subunit. So we have a genetic issue that affects the production of the alpha subunit. It's most common in Asian and African demographics, and it's easy to remember because alpha, Asian, African, and can present with 4 different alleles. So what I mean by alleles is possible combinations. You can have a single deletion or single gene deletion, and that's a silent carrier. Silent carriers are asymptomatic. With 2 deletions, you can have alpha thalassemia minor, mostly asymptomatic, but can be symptomatic in periods of stress or when the body is overwhelmed. With 3 deletions, you have alpha thalassemia major, and most patients would be symptomatic, but overall good prognosis. And if you have deletion to all 4, then that is called hydrops fetalis with HB Bart's, and it's considered unsurvivable. And so when it comes to diagnosing this, you need DNA sequencing of the globin gene to make a conclusive diagnosis. We'll talk about that when I go over to beta thalassemia as well. In terms of treatments, as I mentioned, if you don't have any symptoms or if you're a carrier or minor, you don't need any interventions. However, if you have alpha major, close monitoring and iron and blood transfusions. The one thing we worry about is, remember, this is an issue with the protein subunits, right? So the alpha chain, not iron. So you run the risk of overloading them with iron. And that's something we're very mindful of. And if that does happen, we need to consider iron chelation therapy to lower the iron. Hydrocephalus, as I mentioned, unfortunately is not survivable. So all of them require genetic counseling. This can be quite complex. And I talk about cis and trans here. So what I mean by that is when you have deletion to two chains, you either have the choice to affect, when you have two deletions of the 4 alleles, you have an option of affecting both alleles on one side or one on each side. So the cis is when you have both on one side, trans is when you have one on each, and trans is considered usually, trans is considered to have a better outcome, although that's very niche knowledge that's not expected of you. Next, let's talk about beta thalassemia. So as you've probably guessed, you have an issue with the beta chain. And this, instead of 4, can present with 2 alleles, so either major or minor. And because we have an issue, so the specific thing about beta thalassemia is when your body can't produce a beta chain, it kind of is a bit shocked, and it starts producing chains apart from delta chain that it can normally produce. So let's look at this. So you have 2 alpha and 2 beta chains normally. If your body has an issue with the production of just this, it can switch over and produce the HbA2 hemoglobin molecule. And that makes sense because you have 2 alpha, 2 delta, and your body can produce both of these, right? And therefore, what we see when we perform something called hemoglobin electrophoresis is a high level of HbA2. And this is something we can measure. So as I said, it usually makes up 2 to 3%. But in patients with beta thalassemia, this number will be significantly higher. Now, I mentioned that to diagnose alpha thalassemia, you require DNA sequencing. Well, why can't we just do the same thing that we did here and come to that diagnosis? Well, if you look at this, you don't really have another option, because if you have knocked out the production capacity of alpha units, you can't switch over to something that doesn't contain alpha, because all 3 of these contain alpha, which is why when you have hydrops fetalis, where you have 4 deletions and you cannot produce the alpha subunit, you cannot produce any hemoglobin, which is why it's incompatible. And treatment is pretty similar. So with beta thalassemia minor, no specific interventions. With major, close monitoring, transfusions, and iron chelation therapy if required. Now, what an FBE would show, so it would show a microcytic hypochromic anemia. And if you do a blood film, you'd see these things called target cells. And it seems like a target. That's the only way I can describe it. And these other cells called teardrop cells. And this is often due to just the poor structural integrity of the cell. Moving on to iron deficiency anemia, we have an iron deficiency. Well, that can be because of a whole range of issues. A good way to think about it is decreased supply and increased demand. So the most common cases that are important to talk about is issues with diet. So being on a vegan diet, as you know, vegetarian food generally has lower levels of iron. You also need to know, when we talk about iron in the diet, there are 2 types of iron. There's iron that's bound to hemoglobin or heme iron, and then there's non-heme iron. Obviously, a vegan diet will have no heme iron because plants don't have hemoglobin. And that's slightly problematic because your body is way better at absorbing heme iron than it is non-heme iron. You can also have people with gastrectomies or bariatric surgeries who might have issues with absorption, as well as inflammatory conditions like celiac disease or IBD. The next thing is increased demand. Now, what can increase demand? Pregnancy is a big one because you are growing a baby inside you. You can also have increased blood loss, which may be pathological, like a GI bleed, or part of normal functioning with menorrhagia, which is a big reason why most teenage girls are either borderline iron deficient or are iron deficient. Now, how can we assess this? obviously we want to firstly examine for signs and symptoms of anemia, but then also looking specifically at things that might indicate iron deficiency as being the most likely cause. So let's look at this. So in terms of signs and symptoms of all anemias, you can have fatigue, weakness, shortness of breath, headache, difficulty concentrating. So a lot of vague signs, it can be a lot of things, which is why you always want to have anemias in the back of your mind. And pallor of conjunctiva and the palmar creases are a non-specific sign. In terms of IDA-specific stuff, you have this thing called pica, which is the craving for inorganic substances. So I really want to eat ice. I really want to eat dirt, clay. Ice by far is the most common one I've seen. I've met a lot of people who have cravings for ice, and yeah, and a lot of them I would expect to be iron deficient. In terms of signs, you can have cholonychiae, which is the spooning of the nails, glossitis or inflammation of the tongue, angular chelosis, which often overlaps with glossitis, and you can have angular stomatitis, and then also brittle and thin hair. So a few examples: so pale conjunctiva, you can see clearly there's widespread pallor of the creases, but also the entire hand. Here you have angular stomatitis, and then you also have cholonychia, which is the spooning of the nails. The conclusive test is obviously going to be iron studies, and this is really important to understand. Here's a lovely, lovely table that will get you through so much, but please understand this. So what do we look at when we order iron studies? Well, we look at serum iron, we look at ferritin, we look at transferrin or total iron binding capacity, and then we look at transferrin saturation levels. So let me break this down to you and explain this with the help of an example. Let's say that we have an airport and we have a hotel. Between the airport and the hotel runs a bus service that picks people up from the airport and drops them off to the hotel. The transferrin is going to be the bus. And as the name suggests, it transfers iron. Your ferritin is going to be your iron storage or the number of people at the hotel. Your total iron binding capacity is going to be the amount of iron that the buses can carry. And then your serum iron is going to be the people who are just somehow, instead of catching the bus, they just decided to walk the way to the hotel, as stupid as that sounds. And then transferrin saturation is the number of people on the bus. So let's talk about this. Ferritin is going to be, so let's talk about each individual case and we'll talk about how that reflects. Okay, iron deficiency. The first thing is you do not have iron in your body. Your ferritin, which are the iron stores, are going to be low because people are just not visiting. Either people are not visiting or they went somewhere else, but the number of people in their hotel are going to be low. Serum iron is also going to be low. However, serum iron is very unpredictable. It's not a good indicator of your body's iron status. People may choose to walk to the hotel. Sometimes it has a lot to do with the meals you eat and can spike irregularly. We don't want to look at serum iron. Your total iron binding capacity is going to be high, right? Remember, your body's pretty stupid sometimes. If it sees a low transferrin, it may think, oh, well, it's because maybe the buses that are meant to be bringing the people are not being effective, which is why we don't have people at the hotel. So what they say is, well, let's increase the number of buses to the airport because that might fix it, right? Because your body can't control how much iron it gets. It can just control what it does with it. And then transferrin saturation, or the number of people on the bus, is going to be low. Relatively straightforward. Next, we have thalassemia. Thalassemia is, again, an issue with hemoglobin, and therefore iron studies will be plumb normal. You should not have issues with iron with thalassemia. You, however, can have high levels of ferritin if the patient is iron overloaded. Anemia of chronic disease. Now, this is an interesting one. Now, why do you get anemia in chronic disease? So what you need to know is that ferritin, which reflects iron stores, is an acute phase response marker, right? and it goes up in levels of acute inflammation, but does not in chronic disease. With chronic disease, not only do you have a level of inflammation, but you have an issue with the actual iron absorption in the gut. A good mnemonic to, actually, no, that is not a good mnemonic, but to remember where each substance gets absorbed, you have IFB, and that is iron in the duodenum, folate in the jejunum, and then B12 in the ileum. Now, when you have anemia of chronic disease or a widespread level, or if you just have chronic disease and you have issues at the systemic level, your body is not going to be absorbing a lot of iron, and so you might have actually low total iron binding capacity, which causes the low ferritin. As I'm sorry, I realized I've confused you a bit. Let me take a step back. You have a chronic disease. The chronic disease affects the ability of your gut to absorb iron. This is because you have a low total iron binding capacity. Your hotel is not sending enough buses to the airport to pick people up. I also mentioned that ferritin is an acute phase reactant, which means it goes up when you have inflammation. So your ferritin may actually be normal or high, but this is not a good representation because your ferritin, in this case, is high, not because of the iron stores, but because of the inflammation, and your serum ferritin would be low. With iron overload, serum iron is going to be high, ferritin is going to be high, total iron binding capacity is going to be low, and the saturation is going to be high. So again, you are now getting too many people coming to the hotel. The hotel is fully booked. It's overstaffed. It's so booked that people are having to, there's such a demand that people are actually walking to the hotel. It's flowing and leaking into the blood. The total iron binding capacity is now going to start to drop because your hotel is like, well, I can't have any more people in here. Please stop sending more buses. So it reduces the number of buses. And then the number of people on the bus is going to be super, super high because they're fully booked. Acute phase response, again, you might have a lot of things, but the one I want to focus on is ferritin that's going to be high. How do you treat this? Well, you treat the underlying cause, that is, whether it's a supply issue or a demand issue. You want to supplement iron, starting off with oral, then IV, then IM. So oral and iron, then IV if severe. You can also give transfusions if it's life-threatening. As I mentioned earlier, we may worry about iron overload if you're giving too much, and consider iron chelation therapy like desferoxamine. Okay, now let's move on to, oops, did I skip a slide? Okay, sorry. Now moving on to normocytic causes of anemia. Firstly, we have aplastic anemia. So aplastic anemia is a disease of the bone marrow. You are not producing cells, right? This because obviously your RBCs are being produced in the bone marrow. Along with this, we have all cell lines being affected, but because your bone marrow produces not only RBCs, but also WBCs, platelets, everything's shocked. So you have anemia, a leukopenia, and a thrombocytopenia. Causes include medications like chlorphenicol, chemicals like benzene, radiation, hepatitis B, all can cause aplastic anemia. In terms of a biopsy, you would see hypocellular bone marrow with increased fat spaces. As you can see, this normally should be filled with bone marrow cells that are very metabolically active. However, they've been replaced with fat cells, and you have a very low density. Acute blood loss. Now, this is relatively self-explanatory. Why you are bleeding depends on obviously what's going on. But a few common sites that you need to know are mostly GI bleeds. So GI bleeds are a big one and can cause anemias really easily. We worry about them a lot. This is obviously apart from the traumatic injuries that you may encounter during your practice. So upper GI bleeds are most common ones, are bleeding peptic ulcers and variceal bleeds. With lower GI bleeds, the ones we worry about are cancers and IBD. Now, I want to stress that Most upper GI bleeds are more acute and more sudden and worse off in terms of how much blood you're losing. Lower GI bleeds are a bit less severe in the short term, but over time can definitely cause an anemia because you have low levels of bleeding going on for a long time. To manage acute blood loss, well, you would treat it by giving them more blood. And a good estimation of how much blood to give is depending on the degree of their anemia. So we do that, we measure their hemoglobin. We usually have a target of about, depending on whether the issue is a variceal bleed or not, about 100. If it's a variceal bleed with chronic liver disease, our aims are slightly low. So usually 80 to 90 is acceptable. And you should know that one bag of blood will bring their hemoglobin up usually by 10 grams per deciliter. This is important for two reasons. First, it helps with dosing. How much blood do I give them? Well, here you go. And secondly, when you do give them blood, you can kind of monitor how they respond to it. So if you give someone blood and their hemoglobin keeps on dropping, it probably means that there is still ongoing bleeding that you haven't addressed. Next, we have hemolytic anemias. So hemolytic anemias, destruction of RBCs, and you can classify them as either intravascular or extravascular, depending on where this destruction is going on. The two most important extravascular sites are the liver and the spleen. In terms of the other differences, you can have congenital causes like G6PD deficiency, important to know about, and that's typically intravascular. And some acquired causes are MAHA and I don't know AHA. So MAHA stands for mechanical something hemolytic anemia. I'm sorry, I was just blanked. And then you have autoimmune hemolytic anemia. Oh, I should know this. I'm sorry, this is being recorded at a very late time. And some congenital causes of extravascular hemolysis are sickle cell anemia, as well as things like hereditary spherocytosis. You can also have infections like malaria, but I don't think that's super important. What is G6PD deficiency? It's an X-linked condition affecting mostly Mediterranean populations. You have a deficiency in G6PD. Now, in terms of understanding how this is going to present both on blood films and clinically, you need to understand what G6PD does in the first place. So, usually with any cell, you have a process called oxidative damage. What is oxidative damage, you ask? Well, oxidative damage or oxidative stress is a process that each cell undergoes when it's exposed to oxygen, and the oxygen in the cell reacts and produces these things called free radicals. Now, these free radicals in most cases are non-pathological and don't usually do much unless you have a problem with removing them. Ideally, G6PD removes these free radicals, and then and usually you're all good, right? However, when your body is under high periods of stress or oxidative stress, you can become symptomatic. So the most common buzzword on most exams is fava beans that can trigger G6PD deficiency. What is going on here? So your RBCs sustain oxidative damage. However, because you are no longer able to clear these free radicals, they undergo premature cell lysis or destruction. A common buzzword are Heinz bodies. Now, what is Heinz or what are Heinz bodies? If you look here, these blue dots at the periphery of the RBCs, which, as you can see, the RBCs are quite spherical. This is not normal. These peripheral bodies are called Heinz bodies. What are Heinz bodies? As I mentioned, where is the oxygen going to be? Well, bound to iron. Where is iron? It's in the hemoglobin. So the hemoglobin starts undergoing oxidation, and the oxidized and denatured hemoglobin, which obviously is now going to be non-functional, starts accumulating in the periphery in the form of these bodies called Heinz bodies. Now, these Heinz bodies will circulate throughout the body, and one of the places they will enter is the spleen. Now, the spleen is called the RBC graveyard, and a big part of what it does is pick up defected RBCs through macrophages. Now, this RBC is going to enter the spleen, the macrophage is going to notice the Heinz body and is going to try and bite it off. It's either going to try and bite off the Heinz body or it's going to destroy the RBC in its entirety. When it bites off the Heinz body, it leaves what quite literally looks like a bite taken out of the RBC. And these are called bite cells. You also would obviously undergo quite a bit of lysis, and that is going to lead to the anemia seen with G6PD deficiency. Next, we have sickle cell anemia. Now, this is an issue with hemoglobin, typically found in sub-Saharan Africans. The mutation results in the production of a hemoglobin called HbS. Now, its heterozygous form protects against malaria. So we'll talk about why this is the case. Normally, one of the amino acids in the polypeptide chain of hemoglobin is glutamate, which undergoes a mutation with sickle cell anemia to valine. So glutamate to valine is the mutation we see, and valine is hydrophobic. Again, this is not stuff you need to know, but the next time you think to yourself, why does sickle cell anemia present with sickle-shaped RBCs? This is the reason. What happens is you undergo this transformation, and during the process of maturation, this transformation results in the abnormal shape that we see here. Now, well, why do you get anemic? Because again, all these cells will be traveling to the spleen and be undergoing hemolysis, because these are poorly shaped cells and are quite poor in their functioning. Now you have a high level of blood flow to the spleen that's carrying along with it all of these cells that are getting destroyed and stuck in the splenic architecture. And that leads to splenic infarcts. Over time, your spleen's function starts depleting. And what we see towards the end is this thing called hyposplenism, where you've pushed the spleen far beyond its capacity and now it just can't function. You then see these things called Howell-Jolly bodies with sickle cell anemia, along with hyposplenism. What are Howell-Jolly bodies? Now, again, this is so extra, but if anyone's keen to know, this is what's happening. During the process of RBC development, you have late erythroblasts. Now, erythroblasts are the cells that become erythrocytes. As with any cell, you have a nucleus. However, normal RBCs don't have a nucleus. They don't have a nucleus because you don't want to be using any of the oxygen that the RBCs are carrying, right? Because within the nucleus, you have aerobic respiration. We don't want that, so we just get rid of the nucleus. This is usually expelled out. However, if you don't expel the nucleus, you will basically end up with this body on a blood film called a Howell-Jolly body. Now, this is central compared to our friend, the Heinz body, that is seen peripherally because, well, the nucleus is more central, right? Also, a big part of where this removal process happens is the spleen, right? Now, usually the spleen is kind of this checkpoint marker, which would, in most cases, remove these RBCs. However, because now the spleen is not functioning well, we end up with these Howell-Jolly bodies being seen in the peripheral blood. And this is abnormal and seen commonly with sickle cell anemia that's caused hyposplenism. You can see this exact same thing in people who have had their spleen taken out. In terms of intravascular hemolysis, you can either have immune or non-immune, depending on, obviously, the mechanism. With non-immune hemolytic anemia, you have some issue in the structure of the vessel that predisposes the RBCs to damage. Now, this can be a mechanical thing, like a heart valve, and every time blood flows through that, the RBCs undergo shearing and damage. Or it can be smaller things that are more widespread throughout the circulation, and this is called microangiopathic hemolytic anemia. That is what it stood for. MAHA, from 3 slides ago, stands for microangiopathic hemolytic anemia. Now, this can be due to things like hemolytic uremic syndrome, DIC, TTP, malignant hypertension. You don't need to know the specifics of this. What basically is going on here is you have damage to the endothelial layer, which is quite widespread. This causes fibrin deposition and platelet aggregation. And when the blood flows past this irregular platelet aggregation, you're going to have hemolysis. Now, with immune hemolytic anemia, you have complements, your C3 and your C4, which attach onto RBCs, and those eventually are, because of this attachment, they are marked for destruction and get destroyed. Now, you may be, well, why are we having this complement fixation in the first place? We don't know. In a lot of the cases, it's actually idiopathic, and we don't know why that's going on. So, with autoimmune hemolytic anemia, this can also be extravascular, but it's more rare. What's going on is, again, macrophages in the spleen would phagocytose and lyse the RBCs that are bound to antibodies. The test to do for these cases is called a Coombs test, and we'll talk about what the Coombs test is in a second. Whenever we have antibody-coated RBCs, it can undergo one of two things. As I talked about earlier, with these damaged cells, you either take the entire thing out, or you take a bite from them, or you remove a part of them. And so either you have just no RBCs, which is the anemia side, or you have these poorly formed RBCs in the form of serocytes. Now, what is the Coombs test? So the Coombs test is a wonderful test to look for autoimmune hemolytic anemia. What's going on here? So let's say you have this patient A. He's going to have RBCs, right? And now they have antibodies for these RBCs. We take their blood and we put it in a test tube. For these cases, we know what the antibodies are. And because we know what the antibodies are, we can, in the lab, generate antibodies to antibodies. Let me say that again. Because we know and have studied these antibodies, we know what they are, and we can generate antibodies for these antibodies, right? And this is called the Coombs reagent. When we add this, it kind of links all the antibodies together, and the antibodies are linked to the cells, so it's kind of holding everything together, and you would notice that the cells clump up at the bottom, and that's a positive test. The other thing you can do is, instead of taking the blood, which already has RBCs bound to it, you can take the patient's serum, which contains the antibodies. Again, obviously, this is not going to be bound to the RBCs just yet. We then give or we use a donor blood sample to add the antibodies. And once these antibodies are again linked together, we can use the reagent that we talked about to cause the exact same thing. Now, you may be wondering, Karan, why do we go the extra step and not just take their blood every single time? Well, with autoimmune hemolytic anemia, you not always have this high level of association or linkage with the antibodies and RBCs. It can flare up, and at times it binds, and then you have a drop in your hemoglobin, but other times it's fine. However, they almost always will have the antibodies in their serum. However, the direct Coombs test is the one that's more commonly done, especially in periods of acute flare-up, and is diagnostic for autoimmune hemolytic anemia. And I've already talked a bit about the spherocytes. Now, a hemolytic screen. So these are the tests we order when we suspect hemolysis. So let's talk about what each one of them are and what they mean. Because we're undergoing hemolysis, our hemoglobin is going to be low, right? Easy peasy, that's easy. Then we have lactate dehydrogenase, which is an enzyme used to break down cells. And it's an enzyme that's produced when you break down cells. So when you break down muscle, you release lactate. When you undergo anaerobic respiration, you release lactate. So this is going to be high. Reticulocytes are immature blood cells that haven't fully developed yet. And usually, you should not have a lot of reticulocytes in your blood. However, when you've undergone this hemolysis, you would have high levels of reticulocytes because your body is going to be trying to make up for the RBCs it has lost. Haptoglobin is this neat garbage truck. So whenever you have hemolysis, whenever you have cells that are lysing intravascularly, it sends out haptoglobin to go and collect all this junk, right? So when you do collect all this junk and are now going to the spleen to discard off all this junk, you are not going to have these garbage trucks because these garbage trucks have been so busy cleaning up all this mess, which is why your haptoglobin tends to be low. Your bilirubin or your unconjugated bilirubin particularly is going to be high, as I'm sure you know, and you're going to have a positive red Coombs test. Next, talking about macrocytic anemia, the mnemonic is BUM-HP, B for B12 and folate, A for alcohol, liver disease, myelodysplasia, hypothyroidism, and pregnancy. Now, there are two kind of broad categories we can split them into. We have either megaloblastic anemias or non-megaloblastic anemias, depending on the size as well as the lobularity of neutrophils. We'll talk about this in a second. The reason we get macrocytic anemia is usually to do with the developmental pathway of RBCs themselves. As you know, RBCs, obviously, part of it is going to be forming the DNA and then forming the immature RBC, which has the nucleus in it, and they're taking the nucleus out and changing the shape a bit. So everything here fits in somewhere along the developmental pathway. Right? When we have issues with DNA synthesis, we produce weird-looking nuclei, right? And that's going to cause megaloblastic anemias. Drugs, cancers, and B12 and folate, which you know are important for DNA synthesis, are all examples of megaloblastic anemia. The other thing is the liver has a very important role to play. So whenever you have anything affecting the liver, you can end up with, again, a macrocytic anemia because you have stunted the division of RBCs and their maturation. So now you're going to have bigger cells that are not usually the best. So things like alcohol, things like liver disease are an important one. Lastly, we have hypothyroidism. The reason hypothyroidism presents with the macrocytic anemia is that you need thyroxine for RBC division and maturation. And therefore, hypothyroidism is going to present with macrocytic anemia. Again, you can have megaloblastic and non-megaloblastic. We already talked about this. And a big part of it is going to be the segmentation of neutrophils. Already talked about that. With one, you have arrested nuclear maturation, and with the other, you have abnormal membranes. Does that make sense? Issue with the DNA pathway, issue with the lipids, because the lipids is what makes up most of the cell membrane. Lipid metabolism, quite liver-centric, and then hypothyroidism, already talked about. B12 deficiency, it's most commonly due to either issues with intake. It's almost always issues with intake. You don't really have excessive levels of B12 consumption. The big one is going to be a vegan diet. As I mentioned earlier, B12 is very commonly found in meats, and therefore people with the vegan diet are at a higher risk for B12 deficiency. You can also have impaired absorption. A particular big one is pernicious anemia with atrophic gastritis. Now you have the binding of B12 happening at the level of the stomach, although B12 doesn't get absorbed much later in the terminal ileum. When you have issues with gastric secretions, you can develop B12 deficiencies for this reason. You can also have medications like colchicine, ethanol, metformin, already talked about. Methotrexate is the big one that comes up when we talk about macrocytic anemias, but you should know that methotrexate is a folate antagonist, which is why it causes folate deficiency, not B12. Just know that. You can also have signs and symptoms of peripheral neuropathy that are going to be specific to B12 deficiency. And then you can also have this rare complication called subacute spinal cord degeneration that's irreversible. It's often much, much later. Same principle as peripheral neuropathies, but just now you're affecting the central nervous system. Now, folate deficiency. Now, folate is found in foliage. or green leafy vegetables. The body has a smaller store of folate compared to B12, which is why the deficiency kicks in soon after you stop taking folate, which is usually 3 to 6 months, versus B12, where you have huge levels of B12 stores, usually good enough to last you a few years, which is why when someone switches from an animal-based to a vegan diet, they will not become B12 deficient for at least 2 to 3 years. It is, however, in today's society, very, very difficult to develop a folate deficiency because almost everything we eat is supplemented with folate, even bread. A good example of a common buzzword is someone who's homeless and doesn't eat any vegetables. That's going to be your folate deficiency. Sorry, vegans should not be in there. Vegans are not at risk of folate. I'll take that out later. And this is basically everything that we've talked about. In terms of what to give with folate, we give them folic acid. That's usually oral. With B12 deficiency, we usually give them hydroxycobalamin. The reason we give them hydroxycobalamin is, again, if you think back to what the main causes were, well, we have a vegan diet, but that's not going to be a cause really in most people because it's hard. And takes a long time for a vegan diet to affect B12 levels. But the big one we talked about was atrophic gastritis and pernicious anemia. And that's an issue in the gut. So it doesn't really make sense to be taking more oral B12 because, well, despite how much you take, you're just not going to absorb it, which is why we give it through an alternate route, through hydroxycobalamin that's given intramuscularly. Now, very quickly, I'm going to go through a few blood film findings. So target cells are a finding that's very commonly seen. A good mnemonic to remember the causes of target cells is HALT. And HALT set the hunter to its target cells. Any issue with hemoglobin, so thalassemias as well as sickle cell anemias, asplenism or hyposplenism, liver disease and thalassemia. Pencil cells or elliptocytes are specific to iron deficiency. You can also have acanthocytes, which look like it's meant to be a can of beer. They're called spur cells, typically seen with liver disease, but also hyposplenism. Oops. Howell-Jolly bodies, already talked about what is going on there. This is basically an issue with DNA maturation. So anything affecting the DNA pathway is going to cause Howell-Jolly bodies. And then Heinz bodies are our peripheral oxidized hemoglobin particles, and think fava beans. And that would eventually transition to bite cells. And here's just again a graphical representation of the different causes. Good reference, although it's very unlikely that you'd be tested on specific knowledge when it comes to interpreting blood films. And that's all we had for today. Thank you all for coming. This was quite a long lecture, so I'm sorry I went a bit over time in that regard. But again, if you have any questions, please feel free to get in touch. This is quite a big topic. So please try your best to understand it. And as always, please look after yourself and please look after your loved ones.